So this allows me now to announce the first speaker. Our first speaker is from TU Graz, Paolo Falcaro. He's really the pioneer in encapsulating enzymes in metal organic frameworks for catalytic applications. He received his Master of Science from Padua University in Italy and his PhD in materials engineering in Bologna University in Italy. And now he's full professor of bio-based materials technology in TU Graz. And Paolo, it's a great pleasure to have you here. We are very much looking forward to your exciting presentation. The floor is yours.
Thank you so much.
All right. Thank you very much for this very prestigious opportunity. I'm very thankful to the organizer. And today, it's my task, my pleasure to discuss a little bit about the advancement on ZIF biocomposites. And we will try to have a look, according to my vision, about the past, the present, and the future of these materials. Now, my first important task is to acknowledge that what I present today is the result of a number of great collaborators. And you will see some of those faces around the presentation, but I'm very thankful to the group of Professor Dunan as we work together. We are far apart, but it is actually a progress that we are doing together towards a vision that we share. Then I would like to acknowledge my group. So those are the great people that they progress the work in the lab, because as the most of the PIs, we actually have not much opportunities to get closer to the lab, fortunately for us. Now, the third slide is to acknowledge the funding organization, those that they make financial support, and they allow us to continue our research. TU Graz, that set some money to grow up a community on porous materials, the ERC, the Horizon 2020, and a NATO project. So to put the presentation into the context, we have to define MOF biocomposites. Well, yesterday, Professor Yagi defined MOFs, and who better than him could do that? In this case, we have to go through the different classes that with Christian and Darren defined. And we have MOF biocomposites that are prepared via surface immobilization here. So we stick a protein on the surface, we can covalently immobilize this. We could infiltrate MOF within the pores. This requires the engineering of the system to have pores that can fit this. Or we can go for the encapsulation. And that's exactly what we're going to discuss in the journey today. The encapsulation has an advantage that's unique. Well, we can take a biomarker molecule of any size, and we can grow a MOF by self-assembly around it. And the idea is that once we mobilize an enzyme, the enzyme, if it has a pores of the proper size, can be active, processing substrate to products. But it is expected that the cage around it would protect this precious entity, protect it from proteolytic agents, enhance the thermal stability, and eventually could be used or exposed to solvents as well, right? And at the very end, what we expect is that the enzyme keeps at least part of the activity once it is set back to work. There are a number of different research groups that they try to establish the technology of enzyme encapsulation for different purposes. For example, if we use enzyme, we want it that are expensive biocatalysts, we want to reuse them. And a good way is to stick them on materials. That's how we can lead towards sustainability for this particular aspect. But if you have to prepare pharmaceutical or you have to prepare chemicals, extended use of enzymes that are very precise catalysts would help technology. Now, what are the MOFs that are suitable for this encapsulation? Or better, what are the most MOF that we will discuss today, and you can find also in literature? Well, ZIF-8, ZIF-90, and MOF7. They are all metal isolate frameworks, right? The first is prepared with zinc at 2-methylimidazole, the second, again, with zinc, and the third one with zinc as well. So we have the carboxyaldehyde and the triazole that they play a different role regarding the hydrophilicity and the hydrophobicity. Why are those the best MOFs? Well, there are different reasons that are experimentally related to the conditions that are suitable to prepare those MOFs with our biomacromolecules. But we have to remember that we look for the soda-like topology because we have to select a MOF that has certain porosity. And those MOFs are, especially ZIF-8, is stable in water for a long time, as the paper in PNAS from Professor Yagi disclosed. But ZIF-8 can be prepared under mild conditions, so suitable for maintaining the activity of enzymes, has a low cytotoxicity. We can debate about this, right? But it is considered like that. And it has a stimuli responsive dissolution behavior. And we'll see why this is relevant. So the first approach that was developed for the immobilization of enzymes was using PVP. So polyvinyl pyrrolidone has the ability to collect ions and thus enhance the concentration around enzymes that were stabilized with this polymer. The very first paper that we could find in literature is from Professor Jun Jae. And he used PVP with cytochrome C with 2-methylimidazole. He used methanol mostly. And he showed that this material has an enhanced activity once it goes through the oxidation of amplex red. Importantly, you can see from these scanning electron microscope images, then after treating the sample with high temperature, we preserve the shape of our MOF. So the rhombidodecaedron shape is preserved, but you see all the pockets here, right? Where the proteins were possibly located. Another approach that is very similar, in my opinion, is the one provided by Fa-Quang Shen and Frank Su in JAX. And this is related to the use of PVP in water this time. But using the carboxyaldehyde, a temperature was needed. So PVP was supposed, in my opinion, used to protect the enzyme for the 42 degrees necessary for the dissolution of the ligands. And here they showed that the MOF was acting like a protective sieve to avoiding the contact between protease and urease. Our approach is slightly different. So we were used to take advantage of heterogeneous nucleation. We used nanoparticles in the past in the group of Anita Hill. And we tested proteins as well. So proteins in water, under certain conditions, can lead to the formation of MOF biocomposites. And we can select, of course, enzymes that they lead the diffusion of precursors within the ZIF-8 MOF. Now, the method that we call biomimetic mineralization shows that the presence of proteins, they lead towards the formation of particles. Here, you can see on the left-hand side, the mixing of the solution just with the 2-methylimidazole, zinc acetate, and water. On the right-hand side, we add BSA, which is bovine serum albumin, a very cheap protein that we typically use as a model protein because the cost is very low. So once we add the protein, you can see that the solution becomes cloudy, right? And it takes 60 seconds. Within 30 seconds, it is very milky. So this shows the ability of proteins, not all, but most, in seeding metal organic frameworks, in this case, ZIF-8. Now, if we separate the biocomposite, we can see the truncated octahedrons via SCM. We can see that the diffraction pattern is the same as ZIF-8, but once we analyze with vibrational spectroscopy, we see the presence of the amide 1 and amide 2 bands that are typical of the proteins themselves. Now, if we label those proteins with the dye, polyurethane isothiocyanate, and we examine the crystals with confocal microscope, we see that there is a good signal homogeneity that comes from the different sections of the crystals, showing that the protein was embedded within the entire volume of the crystal. Now, our hypothesis was that the protein was able to attract enough zinc and provide locally the conditions to induce the formation of the MOF. If we put with another schematic the protein and the ZIF cages within the same scheme, we can see that there is no way we can infiltrate the protein within the crystal. But what we miss from this scheme is the small empty pocket that plays a very important role. So this empty pocket was discovered when we were at the Australian synchrotron, and we use a small angle X-ray scattering. And what we defined with this was the radius of gyrations of the objects that we have within the MOF. So this means that we can define the radius of gyration of the protein, but also of a pocket that was actually slightly bigger than the protein, between 15 to 30% bigger. Why is this relevant? Well, while trying to discover what could be the mechanism of protection, we actually made a correlation by different mesoporous materials and bioconjugating horseradish peroxidase, boiling this in water for 10 minutes, and define that the free enzyme dies, no activity. And while choosing smaller pores, we could actually find out that the pore correlates very well with the residual activity, and the higher activity is provided by ZIF-8. So why is that? Well, if we actually refer to the protein itself, the tertiary structure is well protected by a tight pore, right? So this is very intuitive. So with this, we could define that this system was actually suitable for the protection against different environmental stressors, from proteolytic agents to mechanical stressors. Now, this is a very important part of this research. But if we try to extend the value of what we have in order to gain the opportunities from other fields, we can look at biomedicine. And after reading this Nature Review drug discovery paper from Bob Langer, we got this idea to see if we could actually deliver proteins as biopharmaceuticals, because the statements suggest that using biotherapeutics could actually enhance the potency when compared to small drugs. But again, the issue is temperature, right? We know this with COVID vaccine, that initially the problem was through the storage of the vaccine into suitable temperatures. But we have no idea at this stage if we can release proteins and those are still active. So what we did initially was to encapsulate protease in one batch and a fluorogenic protein into another batch. and leave those at a pH that was suitable for the decomposition of ZIF-8. So what happened over time under this ideal conceptual experiment should have been that protease, once released, could chop the fluorogenic protein and thus providing fluorescent signal. So we did this experiment and we actually found out that this was working. You can see this red line that is the experiment that we just described. The red line is when we just put the two proteins together and one chops the other one. While the negative controls, they suggest that once they are trapped within the crystal, they cannot emit any light. So this was, in our opinion, a kind of very promising way to consider those materials suitable also for biomedicine. The stimuli to release enzymes and proteins in general, it's not just related to pH, it could be related to chelating agents, but also some buffers, they are suitable for the release of proteins. This could be voluntary or involuntary if you don't know that. And you can check this paper from Jeremiah Gass-Smith. We actually defined that, for example, phosphate buffer saline media, they actually degrade ZIF-8 and they lead towards an amorphous zinc phosphate solid material. But if you wanted to have a better look about this field, I suggest checking this great review from Anna Bunsen. So overall, I presented what I consider, and that's just my opinion, the manuscripts that were leading towards the foundation of this field that expanded in different directions. And at the very beginning, everything worked with proteins and enzymes. Then, as we discussed, we went to therapeutics. But people started embedding nucleic acids, carbohydrates, lipids, more complex entities, cells, viruses. Other aspects that have been investigated in order to provide with this extension were the mechanisms induced in different aspects: formation, protection, release, and also the structure. There are a lot of papers where people try to define different phases, building blocks, particle sizes, and this leads to the knowledge that is needed for the development of more complex systems. Now, within this presentation, we will actually check part of the mechanism. We go through the structure, and then we will move towards a couple of examples on complex entities. Right. So for the mechanism, we try to validate, again, with Professor Dunan, the correlation between the properties of the proteins and the biomimetic mineralization. And we actually found out that when checking the isoelectric point, the electrostatic of the protein was the determinant variable for the formation of the biocomposite. Whatever was below 5, below 6, was actually able to induce spontaneously the formation of the MOF. And this could be also measured with the zeta potential. So if we have something that is below minus 30 millivolts, then we have the natural encapsulation. Of course, chemically, we can also switch the property of these proteins via amination, for example. We can make something that naturally induces biomimetic mineralization into an inert protein within the same conditions, or vice versa. For example, if we use succinylation. So the electrostatic of the protein is actually a very important aspect. And not just with proteins. Here we have a video where we use different carbohydrates. Most of them are large in molecular weights, such as dextran. But the only one that works in this context is the one that has a carboxyl group, so carboxymethyldextran, that worked in seconds, as you can see here. So this was a model drug carbohydrate that we used. Carboxymethyldextran is not really useful for anything like PSA, but this was actually helpful because once tagged with fluorescein isothiocyanate, we moved towards an investigation of the amounts of carbohydrate that could be encapsulated. So first of all, we defined the suitable conditions for the spontaneous encapsulation. We figured out conditions where we could have the 100% encapsulation of this model biotherapeutic. It means that the valuable component is all captured by the MOF. But this doesn't mean that if we have high value, we have also high release. It is possible that within the MOF, you can also originate a coordination polymer where you have your protein or carbohydrate that coordinates with zinc. So only for one specific value, we could find the 100% release. And this is what it is important also to check when you work with these systems. Now, as said, this was a model therapeutic, but then we went to clinical therapeutics and we use the glycosaminoglycans. We use hyaluronic acid, heparin, chondroitin sulfate, and dermatan sulfate. With this, we could test the different MOFs from ZIF-8 to ZIF-90 and MOF7. So this study defines also that in conditions where, for example, we have diamondoid structures, once we add heparin, in this case, we trigger the formation of the less thermodynamically stable sodalite topology. And the same happens with ZIF-90, where without heparin, we have a sort of diamondoid topology, while with heparin, we end up with sodalite. With MOF7, we had a partially crystalline material that became almost amorphous after the encapsulation of this clinical therapeutic. But what we discover is that in this case, and despite the crystallinity, the encapsulation efficiency was higher for the triazole-based MOF in all conditions of the biotherapeutics that we tried. And the release seems to be also a function of the particle size and the amorphous phase. So we had a very rapid release. So this requires further work if we have to use for the delivery of a drug and if you wanted to tune precisely the release rate. Importantly, because heparin is easily consumed in some tissues when we have medical conditions, for example, with a parinase, we studied the protection of heparin to see if it was around or inside or partially a combination of the two into the different MOFs. And we found out that the higher protection occurs for the smaller particles, the MOF7. And this was surprising, but also interesting because the small particles were actually confining all the biotherapeutic inside. So the original recipe was actually a good starting point. Indeed, the ligand to metal ratio of 4:1 was actually considered to give sodalite. But once we move from Australia to Austria and we start playing with the system, some postdocs were actually not really able to come back with sodalite. So why was that? So there was a certain protocol with some tricks in mixing that was working, and this was working sometimes, or the most of the time with some postdocs, but not with all, right? So the first task was to define what are exactly those conditions that allow you to get sodalite when you want sodalite, because sodalite is the only phase that is porous at the moment, starting from 2-methylimidazole. And we actually found out that it's not just the postdoc. or the PhD student. It could be the protein itself, it could be the washing procedure, and some of those, they can affect actually the formation. In any case, from the synchrotron investigation, you start from an amorphous phase. And if you wanted to see exactly this amorphous phase, I suggest to look at this work from Pattenson that shows with TM selecting subsystem the beautiful evolution from amorphous to crystalline using the encapsulation of BSA. So Webin was the one that actually found conditions where we preserve the biomimetic mineralization, but at the same time, we have a bulletproof system. Always you end up with sodalite, no matter the process or the washing that you use. And this was good because sometimes you actually need only that topology. And Francesco, inspired by this study, decided to go through a phase diagram. So he made a number of experiments and repeats of those experiments, maintaining the weight constant between the metal, the ligand, and the protein, a typical type of visualization that you have in material science. And he actually discovered that you have some different phases, some of them that were really unknown, but the protein seems to block the evolution of the topology in some metastable phases. Now, once washing the same samples with ethanol, what you could see is that you have also phase transformations. Why is this important? Well, it is important because once you characterize your sample, typically if you do SEM measurements or you want to measure with the XRD, you might wash with ethanol. And you measure the property of the material, and then you characterize a different material, right? So this provides you with the idea that washing procedure, especially when you have this new phase that we actually characterize, and you will see later that was named ZIF-C, is actually very important not to expose this to ethanol. Now, those different phases, we know that a diamondoid was discovered, sodalite was discovered. We could find conditions where we have calcinite as well, that was produced only with mechanical synthesis. And we got this ZIF-C that, according to the vibrational modes, contain carbonates. And then we had something that is mostly a combination between zinc and the protein, so we cannot see effectively the ligand in the solid material. Now, the ZIF-C was intriguing because we got such a huge, large area for this. And we asked Professor Zhu to check and to help us. And she ran as an expert all the TM characterizations. And she actually found out that this was a phase that they had already published before. And this phase was prepared in DMF, presence of CO2 and high temperature. In our case, just pure water was enough to collect CO2 from the atmosphere and making these precursors available to be integrated into the network and giving rise to a non-porous MOF. But other phases that have been isolated and they require not a lot of material to be characterized. For example, here we have a work that shows how Zehao Wang managed with the same methodology to characterize and to define the crystalline structure of U unknown 14 that is now called ZIF-EC1. But if we go back to our original aim, is checking those phases and looking at the properties, we can see that 3 of them are actually relatively good for the encapsulation efficiency. And we said this is important because we wanted to keep all the important components into our MOF. They have different release rates that could be attributed to the bonds that we have and the density of the material and the particle size as well. But in general, we have trends that suggest how one phase could dissolve faster than another. For example, ZIF-C is always faster than sodalite and diamondoid, regardless of the particle size. And using a clinical therapeutic with the same isoelectric point, we could reproduce the same results and more or less the same release profiles. So for those that they don't want to spend time with the characterization of the phases, there is a paper that has been arranged by Michael, Laura, and Francesco, where you have an app, and now we have seen that the app are important this morning and yesterday, where you can define exactly what is the percentage of the different phases within your biocomposite, so you don't have to waste time to characterize your diffraction patterns. Now, one question that you might have is, what is the cost and the balance between the MOF and the therapeutic, right? So here we made a table with Miriam and with Mercedes, where we use the different encapsulation efficiency or loading capacity that we found different manuscripts. And we see that, for example, referring to our test material, this is actually valuable because for the cheapest clinical therapeutic, the cost of the matrix that protects it is 3.2%. If we use an expensive biotherapeutic, then could be the biotherapeutic could be 10,000 more expensive than the matrix itself. So if we can protect and maintain an additional 50% of the activity, this is a good value, in my opinion, that could be added to the pharmaceutical that we are protecting. For a case that we all know, which is insulin, we see how ZIF-8 is 0.1% of the cost of the biotherapeutic according to the loading that we got. Now, there are different groups that have been working. I have to select, for example, 3 of them that they show different type of applications. There is the group of Singamaneni, where they could encapsulate biomarkers, and that's very important because biomarkers, typically from this manuscript, they grade within 2 hours. So if you are in a region where you cannot have immediate contact with an analytical facility, you can actually use ZIF-8 as a dry ice to keep these biospacement. Itamar Wilden managed to co-encapsulate insulin and glucose oxidase that in presence of sugar can transform glucose in gluconic acid. We have a drop in the pH and we have the release of insulin. And Shenqin Ma encapsulated monoclonal antibody showing how this could be preserved against temperature, solvents, and mechanical stresses. Referring to the antibodies, this is how we start now discussing, and we move from the mechanism structure, we go to complex biocomposites. And that's one of our latest results that we published. So here we take antibodies and we add into a solution where we have metal cations, zinc, and D2-imidazole. And we actually see that we have deformation of ZIF-C. And once we dry the sample, we have a stacking of layers. But all of them, they have a specific holes if we burn the material and we examine these with atomic force microscopy. So we went again to the synchrotron. We like to go to the synchrotron and we use a stop flow system where we studied the formation of this biocomposite. Now, what we observe is that with time, we have deformation that is first related to particles, and then we have plates. And we actually define that the material starts forming around the fragment crystallizable region, which is the highly conserved one. It's not the one that binds to antigens. It's the one that is typically used for the bioconjugation, and the immune system see as a flag to target the enemy.
Check, 1, 2.
So what is the implication of that? Well, just mixing this stuff together, we ended up with the plates, where those plates, they have a very high efficiency in bringing the fragment antigen binding able to target the antigen. So with this,
Hey,
we had to, it's not really easy to actually
1, 2, 3, 1, 2, 3, check, check.
And what we did
Check,
was
check.
to actually chop
Hey,
the
1, 2, 1, 2.
antibody in different fragments.
Hey,
We chop and we bought
hey.
those fragments,
Hey.
and we actually found out
Hey.
that the only part that was leading towards the
check.
same morphology was the FC part. So this part, this experiment suggests that the MOF grows in the FC part. But we wanted to see the property, so we tagged with electrofluor the FC part with the aim to create a fluorescent particle and to target the proper cancer cells. So we select the proper antibodies for the proper cancer cells. And here we have a local investigation. So it's an optical confocal microscope that shows if we have the right antibodies for the right cell. We have a precise targeting that is very efficient. If we either swap a cell that has nothing to do with those antibodies, or we use the wrong antibodies, the unspecific binding is not really working well. And the statistical investigation is actually coming from this luminescent measurement. So we made a step ahead, and the idea was also to combine nanoparticles to see if we could end up with the same properties, with the same plates, in order to increase the complexity of what could be used for potentially therapeutic or imaging as improved or new nanoparticles. So for the electrofluor functionalized FC part, we also had a quantum dot. And the idea was to target those cells in the same way. And we actually ended up with similar results. So we have the luminescent signal that is coming exactly from the same spot at the same time when we have the proper antibodies for the proper cells. So the unspecific binding is not happening in a very substantial way. And the vision is this one. So one future goal related to this material is that if you are at the point of care and you use an easy machine, because we're not talking about a complicated machine, we are talking about pumps, right? Syringe pumps that have to combine components together. There is the chance to add either a cocktail or apoptotic agents that are more suitable for a certain type of disease, or maybe we target this cancer or bacteria with specific antibodies. So this is where I hope one day someone will lead some substantial results. Another field that I found very intriguing, and it is part of the complex biocomposite examples, is the growth of these MOFs around shells and viruses, about cells and viruses, creating a protective shell. And here is where we pioneer some of this work with Professor Dunan and Professor Caruso using yeast. But this was expanded and worked pretty well for viruses. And this is work from Jeremiah Gassmith. And recently, Professor Don Pio Kim managed to coat mammalian cells. So this is relatively interesting. And the reason is that we can create around those fragile entities. Here we use yeast cells to get a protective shell that is rigid. So the cell cannot replicate. It is frozen there, but we can let nutrients going through while we can protect the cell from large apoptotic entities, right? And the good part is that once we don't need the shell anymore, we can release the cells are alive, and the cells start reproducing. So here we have an example where we can set the yeast within lithic case, and here we protect these And we can see that the release after 3 or 24 hours in presence of this apoptotic agent that chops the membrane of the cell shows a very high viability. Now, the recent work that we have done was to combine the different phases for the protective shells. And here we have an example where we use the first recipe where we induce the formation of ZIF-8 around it. In the left-hand side, you can see some SEM images where we have some cross-section made with a focused ion beam. So we can see the cell, we can see the coating. The coating is based on ZIF-8, and we could use this system to immobilize proteins such as PSA or antitrypsin. Now, we found conditions where we could have both phases, ZIF-8 and ZIF-C, and we could have a second shell, for example, controlling the second phase. So again, here we can see the SCM, and here we see the cross-section. It's very nice that with the FIB, you see the 3 layers. 3 layers where we have ZIF-8, where we have the protein, and where we have the ZIF-C. So the condition that we have here, where we have the protein, and here we will see the confocal images where we scan through the cells, and the protein is located into the external part. Once exposed to an environment with trypsin, which is a cytotoxic biological entity that will chop the cell, and this is present in inflamed tissues, well, we could release the antitrypsin, and the release rate depends on the external shell that we select. But the antitrypsin is active and inhibits covalently binding trypsin, and thus it mitigates the environment that you have over there, allowing for the replication. The replication study is here, where you can see that this is a measure that shows the opalescence and how it increases with the increase of cells in your system. So the absorbance grows up with the amount of cells. But if we expose those cells prior to trypsin, we kill the most of them. So the delay between this growth is shown by the starting number of cells that in one case are killed. And the same happens, sorry, I always go with the wrong one. The same happens if we use antitrypsin and BSA. So BSA is not able to inhibit anything. But we use those control samples because it is made on the same way, and we just swap an active protein with an inactive protein. And we see that once we have the antitrypsin that kills trypsin, we always have a fast growth. So here we are at the end. And if we have to conclude this with the initial title, the past is related to the pioneering works where people try to combine MOFs and biomacromolecules to explore an unexplored territory. But then research groups have been focusing and trying to get the best out of different systems, and some were easier, some other were more complicated, but we are at the stage where we can put all the knowledge together, and this knowledge could be used for more complex composites. And that's where I can see the future. A future that could be expanded using part of the knowledge that is developed in the field towards the use of other MOFs. So this paper came out, I found this in Twitter and I checked this a couple of days ago. And this was exceptional because they could grow 100 iron on proteins and the number of proteins. It seems that there is no limit now with the isoelectric point. But there are other systems, and I invite you to check the work, the talk today of Francesco, where there is this new platform with halves that seems to be very important. We don't have metals. It is just an organic material that is porous and self-assemble around proteins and makes enzymes more stable. Right. So many thanks for your kind attention. And
yeah, thank you, Paolo, for this wonderful and very exciting presentation. Really great new insights, an exciting emerging area. I'm so excited. I think you will have many followers in this really exciting area. Yeah, we have time for maybe a few questions. I think there was a pre-registered question by Christian Serr, but I also see a hand up there. Please just, can you come to the microphone? Just go to the microphone in the center, please. Or you can use this one, yeah, please. Just. Please, the microphone here, technician. Can you give us the other mobile microphone, please, technician? Okay, maybe in between, I can start with one question from the online chat from Surav Dutta from IIT Madras. He was asking if you have the enzyme biomolecules more encapsulated in the outer shell, and if there is a leaching process, is there some stability issue, especially if there's a highly acidic medium?
The question is if the external proteins that are released, they can make an issue? Is
right, or if the activity is then decreasing?
Yeah. So let's say if you are not very careful with the test that you perform, this is an issue because you could start releasing or measuring the activity of the surface-bound enzymes and not the enzyme that is inside. And this happened, I think, in many pioneering papers where this was not done because there were not the knowledge about that. Now, what, for example, we do is to expose enzymes to trypsin. Trypsin kills what is outside and actually destroy the active part that is outside. There are enzymes that are actually very good in surviving to trypsin. So you have to always run the proper controls without using the MOFs. But yes, this could be an issue.
Okay, thank you. Yeah, please.
Hello, am I audible? Thank you so much for your talk. I had a question regarding your antibody at ZIF sheet paper.
Yeah.
And so the fluorescent tagging is at the FC end.
Mm-hmm.
So it makes a very convincing claim that the FC is inside the ZIF. But how do we know if the Fab is outside?
Yeah, that's exactly a good point. So the plates, they have exactly the size and thickness of the FC part. So either we have the FC parts or we have the FIB parts, but because there is one hole that is really compatible with the size according to TM, AFM, and SAX. But to define that the FIB parts are exposed, we run the targeting of the cells. So only for those systems where we had the specific antibodies that they target that specific cancer cell, we had the particles that were attached on the surface. So for the high activity that we had, almost 100%, this showed that we have a highly binding activity, right? So the property, the final property where we actually put the system in contact with the target shows that this works, and it works pretty well.
Thank you.
You're welcome.
Yeah, please.
Thanks for your presentation. It's quite interesting. So I have a question. So how can you confirm that your ZIF and enzyme can uniformly, the enzyme can be uniformly dispersed in your ZIFs? Is like, are there any pure ZIF mixture and ZIF enzyme in your samples?
Yeah, this is a very good question. So that's something that we try also to ascertain, maybe making a comparison with PVP and the biomacromolecule. So what we actually found out is that one difference for one system that we checked for the biomimetic mineralization, where the biomacromolecule induces the formation of the ZIF, you take those crystals, you burn at 350 degrees Celsius. So you burn the protein inside, you chop your crystals, you look with the SCM, and you see that it looks like cheese with the holes within the crystal. For PVP, for example, the same type of investigation shows that the holes are close to the surface. So there are ways where you can check it, and electron microscopy is actually an effective way.
Like microscope is like you can see like the samples like around like probably 10 micrometer with a scale bar. So our question, how you persuade people? So your sample is not a mixture. It's like a ZIF, only ZIF and enzyme composite materials.
Yeah.
So
I understand your skepticism, and if you don't trust electron microscopy, I'm not saying that you need to use a TM that it doesn't fit the resolution that you need. You need the proper TM. So you need a TM that can see 200 nanometers hole, and then you can see those holes. Another possibility is that you run a SAX experiment as we did. So if there is not an empty pocket within the material, there is no way that the density of an external protein fits this. So we run all the controls. So you take your ZIF8 and you examine the ZIF8. Then you take your protein, you examine the protein. Then you take the ZIF8 exposed to the protein outside and you measure it. Then you take the biomimetic mineralization one, and you can see that the only scattering curve that shows the pocket is the one that is related to the biomimetic mineralization.
Okay, is it possible to average the properties? Like it's not like, or is it possible to average the properties you test? You got from SAX?
Could you say it once again?
Is it possible you tested the average the properties is something like?
I didn't get it.
I think it's about the average properties, but I think maybe in the interest of time, we have to move on.
Sure.
And I think, Paolo,
can we talk later?
You will be around the whole conference.
Yeah, yes,
and,
sure.
I think there are probably many more questions. And in this exciting new area, I would suggest please approach Paolo directly. You cannot miss him, and enjoy the discussion with him. Let's give Paolo a great thank you for this very wonderful presentation.